The year is 1771. Chemist Joseph Priestley conducts a series of experiments to see what happens when he heats various substances in closed jars and collects any gases coming off. In one of the experiments, he put a lit candle under the jar, but the flame quickly burned out. In Priestley's words, the air inside was injured. Curious what effect this injured air might have on a living thing, he placed a mint leaf inside the jar and left it there for 10 days. When Priestley checked back, incredibly, the leaf was still green. Somehow the air inside the jar was fresh again. Priestley speculated that the mint leaf must be responsible for this transformation. But how? This question remained unanswered until a Dutch scientist named Jan Ingenhousz decided to duplicate Priestley's experiments. Using plants, Ingenhousz witnessed the same beguiling phenomenon. After conducting a series of more tests, Ingenhousz began to realize what was happening. The ability of a plant to transform the air inside the jar only worked if the plant was exposed to sunlight. This was the breakthrough. Together, Priestley and Ingenhousz had discovered photosynthesis, the biochemical process by which plants trap the energy of sunlight and convert it into chemical energy. Photosynthesis removes carbon dioxide from the atmosphere and replenishes it with the gas on which our survival depends, oxygen. The same gas that Joseph Priestley would discover 3 years later in 1774.
The light-dependent phase of photosynthesis involves two groups of light-absorbing molecules found in the thylakoid membranes. Called photosystem II and photosystem I, they are clusters of light-trapping molecules which funnel energy to special trap molecules. When excited, these special trap pigments release energized electrons. An acceptor molecule picks up the energized electrons and passes them down transporting molecules in the thylakoid membrane. Due to this passage of electrons, hydrogen ions accumulate inside the thylakoid. The energy from this proton gradient is harnessed to synthesize energetic ATP. In an all-important phase, the splitting of water replaces the electrons lost by photosystem II's trap molecule. This step is critical because it generates hydrogen ions for the gradient. It also releases the oxygen humans and other animals need to survive. Responding to light of a different wavelength, photosystem I's trap molecule ejects energized electrons. These electrons are picked up by different carriers until they are added to a molecule of NADP. This forms the second important product of the light phase, NADPH.